The first step of the whole process of mitosis is prophase. During this process, the chromosomes begin to condensate, becoming visible to the human eye. The nucleolus is disappearing. Centrioles begin moving to opposite sides of the cell, and fibers extend from the centromeres. Some fibers cross the cell to form the mitotic spindle. The nucleolus is represented by the inner membrane made up by rocks. This is the nucleolus. The centrosomes are represented by those large rocks. This is one centrosome and this is another centrosome. The chromosomes are represented by the small leaves, which are those, and the fibers are represented by grass. The next step is the prometaphase, which is represented over here. The nuclear membrane dissolves. There are the rocks that were here for the nuclear membrane are not here anymore because there is no nuclear membrane anymore, marking the beginning of prometaphase. Proteins attach to the centrosomes, creating the cytochores. Cytochores are on the middle of the leaves, but they cannot be seen. They are like small dots where the fibers connect them. Microtubules attach at the kinetochores, and the centrosomes begin moving. The kinetochores will be on the middle of each small leaf. The next step is the metaphase. The chromosomes are shown to be the most condensed as possible. They are the best. This is the best step to be seen by the human eye with a microscope. Spindle fibers align the chromosomes along the middle of the cell nucleus. They are all lined by the sister chromatids. This organization helps to ensure that in the next phase, when the chromosomes are separate, each new nucleus will receive one copy of each chromosome, preventing any problem that could result in an aneuploidy. If each chromatid is represented by a small leaf. So if a chromatid is not well organized in this step, it could happen something like Down syndrome or Turner syndrome, because if this was more on the left, on the anaphase, they would come together and they were produce aneuploidies. The next step is the anaphase. This phase is the one where the sister chromatids are separate from each other. You can see that each sister chromatid is in one side because one fiber brought one sister chromatid and the other one brought the other one. The paired chromosomes separate at the kinetochores and move to opposite sides of the cell. Motion results from a combination of kinetochore movements along the spindle microtubules. So basically each one attached to each kinetochore and pull them, making them come to the edge. The next and last step is the telophase and the cytokinesis. Chromatids arrive at opposite poles of cell and new membranes form around the daughter nuclei. You can see that the nuclear membrane is back and that the chromatids can't be well seen anymore. Now, after the telophase, which are the chromatids are already on the edge of each cell, you have separation of the cell by centripetal force. This case only occurs in animal cells because the plant cells has cell wall And cell walls don't allow this to happen. You can see that there are two arrows, which is a centripetal force. It will move this way, and occasionally they will become two cells. In plant cells, centrifugal force occur because you need other forces so that the cell wall can be divided.